Welcome. We're so glad you're here with us for today's Harper Lecture. The Harper Lecture series began in 1979 with Hannah Holborn Gray speaking in San Francisco. As the need for critical thought remains apparent in all areas of life, we proudly continue this signature program to bring you stimulating conversations and fresh ideas. Our UChicago community is a global one, and we are thrilled that technology can help us stay connected to one another. As you enjoy today's lecture, please note the following items. Your camera and video will be off. However, we encourage you to ask questions. Please use the Q&A box at the bottom of your screen to ask a question at any time during the talk. Closed captions are available by clicking the box at the bottom of your screen. Click Show Subtitles to enable this feature. If you're having trouble with audio or video, please try shutting down programs in the background, or you can dial in from your phone. Thank you once more for joining us. We couldn't do this without you. Hello, everyone, and welcome. Welcome to today's Harper Lecture, A Genetic Game Changer, RNA Methylation in Medicine, Agriculture, and Conservation with Professor Chuan He. I'm Andrea Hodgman, Associate Director for Intellectual Engagement and Travel and the Program Manager for the Harper Lecture Series. I'm so pleased to welcome you to today's event. On behalf of everyone at the UChicago Alumni Office, thank you for joining us. Please allow me to introduce our moderator. Yamuna Krishnan has been a professor in the Department of Chemistry at the University of Chicago since 2014. She received a PhD in organic chemistry in 2002 from the Indian Institute of Science, Bangalore, and was an 1851 research fellow at the University of Cambridge in the UK. Her research group pioneered the use of DNA nanotechnology to study living cells and taking DNA nanotechnology into the world of precision medicine. She was featured on Cell's Top 40 Under 40 of scientists that are shaping current and future trends in biology, and the LSDP's Top 100 Global Thinkers of 2014. Professors He and Krishnan, thank you both for joining us. Professor Krishnan, I'll hand things over to you.
That's fantastic. It is my absolute privilege to introduce my dear colleague and friend, Professor Chuan He. Professor He is the John T. Wilson Distinguished Service Professor in the Department of Chemistry and the Department of Biochemistry and Molecular Biology at the University of Chicago. His research spans a broad range of fields, including chemical biology, RNA biology, epigenetics, biochemistry, and genomics. His recent research concerns reversible RNA and DNA methylation in biological regulation. In 2011, his group discovered that reversible RNA methylation was a new mechanism of gene expression regulation. And his laboratory has spearheaded the development of many enabling technologies to study the biology of modified RNA and DNA. Professor He received his Bachelor of Science from the University of Science and Technology of China in 1994, and his PhD in chemistry from MIT in 2000. He then trained as a Damon Runyon postdoctoral research fellow at Harvard before joining the University of Chicago as a faculty member in 2002. And he was selected as a prestigious investigator of the Howard Hughes Medical Institute in 2013. Now, apart from being a superlative scientist and generating technologies that have spun out into very successful companies, Chuan is a great mentor and institution builder. And he has an eye for good people. Many of his students have gone on to positions in some of the best universities across the globe. And he has single-handedly built UChicago's chemical biology group to make it one of the top chemical biology groups in the world. I myself was spotted by Chuan in Munich, and then he recruited me here. And of the various choices available to me at the time, I joined UChicago's chemistry department only because they promised that my lab would be next to his. So Chuan, the floor is yours.
Thank you, Yamuna. Thank you for that generous introduction. I have to say that since Yamuna joined our department, our chemical biology started to take off. Before I start, I really want to thank Andrea and Adam for the opportunity and for making this happen. it's really an honor and a pleasure to have this opportunity and share our research with our alumni and friends of the University of Chicago. So considering this is perhaps a mixed audience, I'm going to give a brief introduction of the epigenetics and the type of science we were interested in. And I will transition there to sort of two basic discoveries that led to something potentially have therapeutic potentials. And at the end, I will transition again to our recent work in plant biology, agriculture, and with implications in conservation. So to start with, I think this is something we're all familiar with. We have tens of trillions of cells, each of us. We have 200 different cell types. So we're very complicated. We can do all kinds of amazing things. But to keep in mind that all of us come from one fertilized egg. What does that mean is out of trillions of cells in the human body like me, their DNA sequence is pretty much the same, right? The genetic information maintained the same, yet these cells have same DNA sequence, very different expression, very different phenotypes, behaviors, even their functions. Another example is this identical twin. These two have almost the same DNA sequence as you guys know. Later in their life, they have different health status. They're different in they have different interests. They have different health and age, etc., etc. So what this said is that the genetic information or the sequence of the DNA alone does not really determine the gene expression or the phenotype of an individual or even the cell type, right? So what happens is that in the genome, we have 3 billion base pairs, right? In different cell types in the human, these sequences are the same. However, they are things on top of the sequence that also affects gene expression or the phenotype or behavior or the expression of the cells. Now, one of these is these modifications on DNA or the epigenetics. Right. It's about 2 to 6% of cytosines can be methylated. You can put a chemical group to the cytosine, and this is reversible. Now that mark can dramatically affect gene expression. So what happens in all the different cell types, although these cells have the same sequence, but they have different methylation patterns. So if we look at a single gene expression, we have something like 20,000 genes inside in our cells. When we look at one gene expression, that expression is controlled by lots of factors, but one critical factor is epigenetics. And what that is these modifications on DNA and on histones. Now, if you have a modification on the DNA or this methylation, that will tend to suppress gene expression. If you do not have this methylation, that gene is activated. Now, somewhere in between, you can have a different dose of this methylation that can lead to different extent of gene expression. So think about if you have 20,000 genes, each gene is subjected to this reversible methylation regulation. And in the human, with tens of trillions of cells, although they share the same identical DNA sequence or pretty much, but in front of each gene, the methylation pattern is different. And that leads to the complexity, diversity of these 10 trillions of cells. So this is so-called epigenetics. In the last several decades, people study this, understand this really affects cell differentiation, development, and aberrant epigenetic will lead to diseases such as human cancer, and the therapies developed to target the epigenetics, in many cases to treat human diseases such as cancer. My lab back in 2009, 2010, decided to look into a different direction. We decided to look at RNA. So we all know genetic information flows from DNA to messenger RNA to proteins, and their chemical modifications on DNA and the histones dramatically affect the gene expression in the epigenetic way. What about messenger RNA? The modifications on messenger RNA, and we propose that these modifications could have analogous roles like the counterparts in DNA's histones may also broadly affect gene expression. So if you look at the human messenger RNA, these are actually modifications one can identify, one can find with modern analytical tools. There's a pseudouridine. If you add a methyl group, that's the modification we use these days for mRNA vaccine. I'm going to focus on this modification called M6A or N6-methyladenosine. It's an addition of a methyl group to the common adenosine base. This is the most abundant modifications in all mammalian messenger RNA. Out of all the messenger RNAs, the bases, every 200 adenosines, there will be one methyl group. So in average, every messenger RNA will carry something like 3 or more methylations per mRNA molecule in mammals. For 4 to 5 decades, people know this is the most prevalent modifications in mammalian messenger RNA, and they actually know there's one component, MET203, that instals this modification, but for several decades, people sort of forgot about it. This is all we know about it, and push it to the back burner. Nobody worries about it. Until 2011, my laboratory discovered demethylase. So these are proteins specifically reverse the methylation, take out the methyl, reverse it back to unmodified adenosine. So we identified and discovered 2 different demethylases. These demethylases are very interesting. They all have quite dramatic phenotypes in mammalian development. For instance, if you knock out the FTO, that turns to be lethal in mice and most likely in human. And if you knock out the RPT5, that leads to infertility in sperm development. Because of the dramatic or interesting phenotype, this discovery suggests that the methylation of mRNA may have broad regulatory roles impacting development. We and others identified the methyltransferase complex. It's a huge complex that most likely regulates the specificity of methyl installation to what type of RNA and where on the mRNA the methyl will be added. Most importantly, in the last several years, my lab and other labs identified and characterized a series of proteins that preferentially binds methylated RNA. For the methylation to have biological roles require specific recognition of the methyl group. Turns out there are these evolutionarily conserved proteins. Many of these proteins, their role is to specifically recognize methylated RNA and regulates RNA cellular localization, stability, and translation. So out of these many years of research, it's become a new field of biology. Back in 2011 was just one paper from our lab. And nowadays, there are thousands of papers every year. From all these biological studies, the 2 hallmarks start to emerge on this RNA methylation. One is on cell differentiation, tissue development. Another is on stimulation, signaling, and stress-induced response. So I'm going to give you sort of the basis of the basic discovery part, and in each case, I'll tell you a story that may be related to medicine. So back in 2015, Jacob Hannan and Gidi Richavi's laboratories in Israel made the knockout mice of the methyltransferase knockout mice, basically delete methylation from mRNA and ask what's going on. It turns out these mice died at a very early embryo stage. It turned out if you do not have methylation, the embryonic stem cells stuck. They do not, they can't prime, they cannot differentiate. That turns out to be the case for pretty much all the cell types tissue studied so far. If you get rid of methylation, your stem cell will not differentiate or will have a major defect. The naive T cells will be stuck. You will never get the mature T cells. Skin stem cells will not give you the mature skin. So in all cell types studied so far, M6A RNA methylation is essential or critical to stem cell differentiation. It's critical, essential to tissue development. It was explained by several mechanisms. One of the mechanisms was explained as this YTHDF2 dependent pathway, first revealed in my lab by my former grad student, Xiao Wang, who's now faculty at MIT Broad Institute. What Xiao and colleagues showed is that this is a protein binds hundreds of methylated RNA and mediates their decay. Xiao and others also collaborated and worked out some animal model experiments and showed that if you do not have YTHDF2, cells turn to stuck. They turn to not differentiate to the next stage. For instance, if you have a fertilized egg, for fertilized egg to successfully move on to zygotes and that further development, these maternal mRNAs that defines the initial stage of fertilized egg, they need to be cleared in order for zygotic genes to take over. That clearance is facilitated by RNA methylation, and these methyl groups are grouped by YTHDF2 for coordinated decay. Another example is neurogenesis. You have a neural stem cell differentiates to neurons. During this process, there are cascades of methylated RNA that comes and go, okay? Now the role of this methylation is shown by this cartoon. So you have a specific cell type. Let's say this is a stem cell. Now these mRNA transcripts are critical for cells to stay in this state. But if you want to differentiate to the next cell stage, if you have a stem cell try to differentiate, you have a cell try to change a state, these transcripts need to be cleared in order for other transcripts to take over. That clearance is mediated through methylation and through YTGF2 binding of these methylated transcripts to decay them. Now, in this example, that's what the methyl groups are for. If you do not have methyl groups, these transcripts accumulate, the cells turn to stuck, and they do not differentiate to the final neuron stage. So with this knowledge, I'm just going to introduce you the first sort of story in stem cell therapy. Now, that was a basic discovery we made in 2014. And that turns out leads to potential new ways for bone marrow transplant. I'm not going to introduce too much about bone marrow transplant, except I will say that this is a way to cure leukemia and anemia or severe blood diseases. In this sort of medical treatment, you wipe away the bone marrow hematopoietic stem cells, the stem cells that generates all the blood lineage, and then you find a donor, either a close relative or someone who has a bone marrow hematopoietic stem cell which matches the patient. Now you take the stem cells out, purify them, and then reintroduce them back into the patients, right? And basically you exchange the patient's stem cells with a donor stem cell, and that stem cell regenerates itself, but also differentiates into all kinds of blood lineages. Obviously, the challenges for bone marrow transplant, number one, it's very hard to find a donor. It requires very strict matching of antigens. Otherwise, the stem cell transplanted will start to attack the patient's system. Second is that the rejection has been a problem. Something like 30 to 40% of patients have different extents of rejection. A way to get by is to use cord blood stem cell transplant. Now, this currently accounted for about 20% of US transplants. Now, in traditional bone marrow transplant, it has a strict requirement to avoid the rejection. However, with cord blood stem transplant, you only need 4 key antigens to match. So it has much less strict requirement. And importantly, there's a cord blood banks these days, and it's readily available compared to finding a matching donor. Now, you would ask, if this is the case, so why this is only 20%? Why can't this be 100%? Well, there are many limitations. One of the limitations is that the limited amount of stem cells one can get from one cord. So in general, for adult recipients for this transplant therapy, 2 cords are required to get enough numbers of hematopoietic stem cells, and that can be used for this transplant access. So on this subject, we collaborated with Dr. Linheng Li. He did most of the work from Star Wars Institute. Linheng is one of the experts in stem cell biology, and he spent a long time trying to figure out ways to expand the cord blood stem cell. The idea has been that if we can expand these stem cells, we are isolated from the cords by, let's say, 8 to 10 folds. Now, instead of 2 cords treating 1 patient, we could have 2 cords treat 8 to 10 patients. So Linheng came to Chicago, we had the lunch together, and we started to speculate, because YTGF2 is so important for stem cell differentiation versus renewal. Remember, when we shut it down, it will not differentiate. We wondered, could we turn on and off YTGF2 as a switch to dictate self-renewal versus differentiation in stem cell systems? So we shifted the knockout mice to Linheng's lab, and all these experiments done in his lab. When he knocked down YTGF2 in bone marrow stem cell systems, and it was great that he indeed observed the expansion or increased the number of mouse blood stem cells or hematopoietic stem cells. And importantly, serious characterizations indicate that these stem cells maintain their stemness. They do not have bias later on in their differentiation commitment into different blood lineage. So they basically are just like the normal stem cells. Lin Heng also tested the human cord blood hematopoietic stem cells. So these are the stem cells isolated directly from human cord blood. And indeed, ex vivo, when Lin Heng performed the knockdown, so suppressed expression of YTHDF2 in these stem cells, he observed about 14-fold expansion of these stem cells. So now we have a way to expand the stem cells ex vivo. That's great. What about if you take these stem cells and put them back into the biological systems? Do you see this still function normal? Could they sort of maintain the type of expansion? So in this experiment, you take mice, wipe away their bone marrow, and then you introduce the ex vivo expanded hematopoietic stem cell into this mice, and then you pass on this for 2 generations, and you analyze whether the stem cell difference between the recipients that had this YTHDF2 knockdown treatment versus control. So in this completely in vitro experiments or in vivo experiments, after 2 generations of transplant, Lin Heng's lab observed 8-fold increase of the human hematopoietic stem cell in this mouse model, indicating that this ex vivo expansion does work, and these stem cells can be transplanted into recipient animals, and these cells maintain their stemness and they're fully capable of generating different blood lineage. And after another generation, they're still fully capable of generating blood lineage. At the same time, they also self-expand into more stem cells compared to control. In a sort of a short summary, essentially, if this can be applied in clinic, what that means is instead of 2 cord for 1 recipient, now we can have 2 cord to treat 8 recipients. So Lin Heng and us are working on this. We have potentially set up the clinical trials. Right now, we're testing a variety of different ways to knock down YTHDF2, either with shRNA or small molecules, and hopefully we can move this into clinical trials and eventually benefit patients. What other stem cell systems? This can't be just a hematopoietic stem cell. In general, this should be a process that can be applied to a variety of different stem cell systems. Another system Lin Heng's lab has been working on is joint regeneration, right? We know when we're getting aging, we have joint problems is really because of the thinning of the cartilage. This leads to swelling and pain and all these troubles. So in Lin Heng's lab, the idea is to use mesenchymal stem cell, but in particular, there's a specific type of mesenchymal stem cell that in Lin Heng's lab, they showed that if we can expand this with our strategy, with YTHDF2 knockdown, we can expand these populations of stem cell and convert them into chondrocyte and eventually implant into the knees to generate the cartilage. So right now, Lin Heng Li's lab, in collaboration with a physician scientist in Kansas City, are working on this, and we're hoping eventually we can also get this system work, and one can start to think about other stem cell systems and stem cell therapies in general. So that's the YTHDF2. One sort of a discovery led to opening up of the stem cell therapy. Another parallel of YTHDF2 is YTHDF1 was also studied by Xiao Wang and another graduate students in my lab. They showed this protein slightly different from YTHDF2, the previous protein I talked about. This one also binds to hundreds of methylated RNA, but when this protein binds this RNA, it turns to promote translation. So if you fix this on a mRNA, you can clearly see the translation is elevated. Now, we look at the knockout mice, and we realize in my lab, we tend to use mouse models to examine functions of the proteins. When we knock out YTHDF2, we realize the mice were fine. Within 3 months, we did not see obvious developmental defects. However, these mice exhibit long-term learning memory defects. Later work showed to us in cultured neuron, if we put these mRNAs in with or without tethering YTGDF1, in regular neurons, we did not see difference. However, when we give a stimulation to these neurons, the one with YTHDF1 tethering showed upregulation of translation. So essentially that explained this long-term learning memory defects at the synapse when there's a learning stimulation requires this YTHDF1 to sense that stimulation, upregulates translation of group of mRNAs there, and that's a signaling, eventually neuron keep memory of whatever stimulation. What this study suggested to us, there's another sort of a hallmark of RNA methylation, that's stimulation signaling and stress-induced response. Now, if you think about stimulation, stress, or response to environment, cures, what systems sort of the most important in a mammalian or in animal, in a human? Well, it's the immune system. That led us to start to wonder about the roles of M6A RNA methylation in immune system, and we used the cancer immunology as a model to study this. So previous postdocs, Michelle and Dali, worked on the system. So in this case, they grow the mouse melanoma tumor and mouse colorectal tumor in Y-type mice and YTGDF1 knockout mice. In the Y-type mice, if you implant these tumors, they grow and then they kill the mice as expected. The YTGDF1 knockout mice are very resistant to tumor growth. Surprisingly, they're just very resistant. We showed that this is indeed a immune effect, because if we wipe out the cytotoxic T cells, this effect is gone. So this is dependent on T cell. We also noticed that if you knock out the YTGDF1, T cell itself doesn't really show much difference. Now, there's a little bit of immunology. It turns out the way it works is if you have a tumor, these tumor cells will release neoantigens. So these are the sense that tumor specifically possess and recognized by dendritic cells. So these are the sensors sense the presence of a tumor neoantigens, process these antigens, present these things on the surface to prime and activate T cells, and these activated T cells come around to eliminate the tumor cells. So if it's not T cell, could this be dendritic cells? The answer is yes. It turns out for dendritic cells to sense the neoantigens released by the tumor cells and process them and then present them on the cell surface, it requires sort of different proteins. And YTGDF1 regulates the expression level of those proteins. In the Y-type, these proteins are presented in the high or translated in the high level. With YTGDF1 knocked out, these proteins are expressed at much lower levels. We can confirm this, these proteins expressed in the lower levels. When these proteins are expressed in the lower levels, there will be more antigens presented on the dendritic cell surface, and that allows more potent priming and activation of T cells to eliminate tumor cells. In the mouse model experiments, we tested the potential synergy with anti-PD-L1 checkpoint blockage immunotherapy. As some of you know, in the last 10 years, the major breakthrough in tumor, in cancer therapy and cancer biology is the development of immunotherapy, right? The immunotherapy works for about 40% of melanoma cancer patients, maybe 15 or less percent of colorectal patients, cancer patients. It works, but it doesn't work for a large amount of cancer patients for a variety of reasons. Now, in this particular case, in the mouse model, you can see in the mouse model, 40% of mice respond to immunotherapy, surprisingly similar to the human case. However, if we combine this with YTGDF1 knockout, we can bump this number to almost 100%. All the mice now started to respond to this anti-PD-L1 immunotherapy. Now, this was great. We sent this paper in, reviewers love it, but there's always a question. If you can cure mouse tumor, doesn't mean this is relevant in human. So we collaborated with our physician scientists here at University of Chicago. One of the major advantages is we're just next to the hospital. We have almost unlimited access to patient samples to our physician experts. In this particular study, we collaborated with Ralph Wassenbaum and Marc Bissonnet. Now, this particular one, we obtained 22 colorectal cancer biopsy samples from Dr. Marc Bissonnet's laboratory or from his clinic. What we looked at is all our studies suggest that the high levels of YTHDF1 suppress immune response. If we get rid of YTHDF1 or we inhibit YTHDF1, we see dramatically improve the immune response. So we look at these biopsy samples on the tumor sites and we ask a T cell infiltration. So if you have more T cells in the tumor site, typically indicates the host will respond to immunotherapy better. As you can see here, out of these 22 patients, we see an inverse relationship as we expected. High YTHDF1 leads to less immune response, and the low YTHDF1 leads to high sort of immune presence or T cell presence in the tumor site, consistent with our basic discovery that YTHDF1 turns to suppress dendritic cells and suppress host immune response to the presence of tumor. Now, with this discovery, we moved on. These proteins have sort of a well-defined pocket. One can think about develop small molecule drugs. We collaborated with another lab. We performed the screening of small molecule libraries. We identified the small molecule is really a natural product, and that natural product can specifically inhibit YTHDF1. And when we tested this in Michelle's lab, she has her own lab now. When we administrate the small molecule to the mice, and we can clearly see the control of tumor growth compared to the control. And again, if we combine this with the anti-PD-L1 immunotherapy, this is without therapy, the tumor grows rapidly. With anti-PD-L1 immunotherapy, with small molecule that inhibits YTHDF1 monotherapy, if we combine the two, we can see the best effect, the synergistic effects of the small molecule with immunotherapy. So I believe these colleagues are currently evaluating this particular compound preclinically, the toxicity and other signs, and we're hoping next year or in the near future we can move this to clinical trials. So I hope I sort of showed you in two examples that some very basic biological discoveries. My lab mostly work on basic biological discoveries, can lead to quite interesting new knowledge, but importantly, this new knowledge learned could impact the patient care in clinic. What I showed on the immunotherapy part is really a tip of iceberg. Together with Ralph Wassenbaum in radiotherapy and other therapies, we show dramatic effect in eradicating tumor, control tumor growth when we target these pathways in general. And hopefully in the next several years, we'll come back and present these results again. And hopefully by then we'll have clinical trials ongoing. Now, with that, I'd like to transition to the last part. My lab works mostly in mammalian systems in human biology, but sometimes what we learned in mammalian system, human biology can have broad implications. And this last one is really a good example of that. So I showed you in the last 10 years since we published our first paper in 2011, lots of people work on this RNA methylation, basic biology, therapeutic potentials. Pretty much everybody work on this post-transfusional regulation. What that means is that once you have mRNA transcribed, the methylation affects the mRNA fate, and how it's translated, decayed, localized, etc., etc. Back in 2018, 2019, a former postdoc in my lab started to think in another direction. So this methyltransferase complex can install methylation to messenger RNA, and June, at the time of the postdoc, started to wondering, could the methylation also occur to these other RNAs on the chromatin? These are non-coding RNAs, they don't code proteins. These RNAs are getting expressed, they stick to chromatin, and people think they notably regulates or markedly regulates chromatin state transcription. And Jun started to wondering whether these things can be methylated and could that methylation control chromatin state? Answer is yes. And we published these results in Science last year, and that was confirmed this year by 3 Nature publications. Not only confirmed our results, but also showed that this regulation is critical for many layers of chromatin regulation. So remember when I was talking about epigenetics of gene expression regulated by many factors, methylation, histone marks. Another factor is this RNA methylation. There are these RNAs associated with chromatin, and they're reversibly methylated, and that dictates the sort of the recruitment or repel of machineries that drive or shut down gene expression. This leads us to think about the demethylase we identified back 2011. So that was the first paper sort of started the field in last decade. We discovered that this protein can reversibly remove the methyl group on adenosine. In cancer cells, we clearly show it removes methyl group from messenger RNA. However, in primary cells tissues for a long time, we do not really know the real substrates of FTO. And this one has a very interesting phenotype. If you get rid of FTO in mice, majority of the mice die at the late embryo stage. Those who are born have clearly developmental defects. You can say they grow smaller compared to the Y type. It turned out in primary tissues, in many primary tissue cell types, mRNA may not be the main substrate of FTO. So we wondered for a long time, what does FTO really work on to impact this development? Jiang Bo, another student now postdoc in the lab, worked on the system and eventually figured out the substrate of this reversible demethylation is LAN1 RNA. So this is a human genome. It's about 1.5% of human genome encodes protein, okay? 20% of our genome is this retrotransposon. This is ancient virus integrated into our genome, amplified itself 1.5 million times. So there are 1.5 million copies of this LAN1. It's a retrotransposon. It can still be expressed. And that RNA from this ancient viral RNA can be methylated. The FTO's primary function during early development in primary cells appears to remove that methyl group. And that turns out to regulate the chromatin state and dramatically affects the mammalian early development. We have a paper under revision. We wish to submit back to the journal in 2 weeks. We worked out how this reversible methylation on this retrotransposon or viral RNA can regulate chromatin state and transcription. With that, we started to wonder and remember, I told you the hallmark of M6A is cell differentiation tissue development. On the mRNA methylation through YTGDF2, I showed you a strategy to expand stem cells. We wonder, could the chromatin regulation also affect cell differentiation early development? Could we modulate the chromatin RNA methylation and control the development? We tested this in different systems. The one that worked out the most is the plant system. It turned out my former postdoc, Guifang Jia, who's a professor at Peking University, had this idea of maybe we can overexpress this demethylase in plants. And indeed, she overexpressed this in collaboration with my lab in rice. And what she observed was this sort of a surprising development of root systems. As you can see, these rice grow roots 3 times deeper and 3 times more roots can be grown with this insertion of FTO to mediate some sort of RNA demethylation in rice. In lab, we were shocked to see the results. In lab with lights turned on and sufficient nutrients, we can grow this rice with 3 times more biomass. You can see there's just a lot more branches of this FTO overexpressed rice, and there's just a lot more yields, about 3-fold increase of yields per rice plant. Because of the deeper roots, this rice more resistant to drought and pathogens. So this is a experiment. We grow them under drought conditions. Now these FTO expressed rice plants they survive the drought conditions better because they have more biomass, they fix more carbon dioxide, and they have elevated photosynthesis. So there's more biomass, there's more carbon fixation. What about in the real fields? So we move this to the real fields. We grow this rice in 2-3 different climate conditions. In the real fields, in the real farm, we saw about 50% increase of the yield and the biomass. You can clearly see this is the one with FTO, this is the control. You clearly see more branch and more sort of grains compared to the control. So I eat rice, and some of you probably don't eat rice. You probably eat potato. Now here's the potato. Again, this is not limited to rice. This is general to many plants. Here's a potato. In the field test, we also observed the 50% gain or increase of the potato production. What's going on? Well, if you overexpress FTO in these rice roots or shoots, if you look at these rice cells, what you notice that they have more open chromatin, as we sort of predicted. When you have FTO there, the immediate demethylation of these RNAs, and that leads to more open chromatin. The direct result of that is if you look at these rice plants compared to the wild type, most of the genes are upregulated. There's less than 1,000 genes downregulated, probably secondary effect, something like 7,000 genes are upregulated. In this diagram, if it's yellow to red, it's upregulated. If it's blue, it's downregulated. By eyeball, you can see majority of these genes upregulated because you have a more open chromatin. What that led to is look at the tip of the roots, the rice roots, they're just the more sort of meristem cells. These are the stem cells that promotes root growth, right? And that sort of leads to the stromatic expansion of the roots. And because you have the stromatic expanded roots, it sends signal to generate more biomass. And what happens is you have more tillers, right? This explains the increased branch and the biomass. If you compare to the Y type, there's just more tillers coming out of this plant. So we're, I started to learn plant biology and agriculture, and I learned some since I saw it. I was not expecting. Many of you probably know in 2030 or 2050, we need increase of food production by something like 30 to 50% in order to keep up a pace with population growth, and that requires a lot of lands, right? We'll have to convert forest grasslands into farmlands, unfortunately. And it was predicted that healthcare has been really advancing amazingly in the last 2 decades. It was predicted in the next 2 decades, the agriculture and the food industry will keep up at the same pace as healthcare, which was a surprise to me. One of my sort of favorite subject or something I really care about is conservation climate change. As I told you that with this expansion of human population, we have to convert valuable lands into farmlands. That means less forest, grasslands that cause all kinds of problems. Already the northern part of the earth was losing these grasslands, forests, or converting to deserts. With global warming, it's just going to accelerate. The question is, can we develop plans to help us to recover these forests and recover these grasslands? There are numerous potential applications: food security, plant stock materials, soybean, corn, wheat. This country, we're privileged that we don't worry about food, but the majority of the world has to worry about food. But we worry about the stock materials come from plants, biofuels, and the part I care a lot is conservation and the sustainability, and of course how we utilize the technology to fix more carbons. So all these questions, hopefully we can have something we can start to investigate, pursue, and eventually develop into real applications. So I shared with you just one modification, M6A. There are other modifications we're currently working on, and at least based on our preliminary observations, there's just lots of new biology remain to be discovered, and there's a lot of potential. Hopefully, we can learn from these systems and apply to benefit not only patients, but also the world. Thank you very much. It's really an honor and a pleasure to give this lecture, and I'll be happy to answer any question. And perhaps we can go back to the Q&A session.
Thank you so much for that fantastic talk. And as predicted, there are a large number of questions. So I don't know whether we will have time to go through all of the questions, but these are really, many of them are very key. So I'm going to encourage you to please stay after this. There will be a small networking session with Chuan so that you can ask these questions yourself, in case I do not ask them. But one of the ones that I found really interesting, Chuan, was that a question from Matthew Nieder, and he says, if such dramatic agricultural improvement can be affected with just the change in a demethylase, why wasn't this found in the standard genetic breeding programs previously?
Right, this is a great question. There are several layers of answer to that. So it turns out what most likely we currently do not really know in the plant system, what's the real substrates of FTO. In mammalian system, we know now. In plant, we predict there's several systems, but the major part is, again, non-coding RNAs. So when we do a standard genetic screen, we really screen this non-coding junk DNA, right? And we think they're like dark matters, nobody care about them, right? So number one, we never targeted those parts. Number two, I don't believe this FTO pathway hits just the one thing. I think it's a combination of several things. So in plant, the DNA methylation is actually facilitated through RNA. It's very different from human. And then in plant, they also have this the plant has a lot more repetitive elements compared to human. That's why the phenotype is more dramatic, we think. And we know FTO also work on small RNA. And again, small RNAs, tRNA fragments, microRNAs, these also dramatically impacts the phenotypes. So there are multiple pathways. We're hoping to, in the next few years, work with elite agriculture lines and to tease this out. For the elite system that has been used for breeding for decades, I don't expect a 50% increase. I'm expecting something like a 15–20% increase for something we've worked on several decades, try to optimize everything, right?
So this sort of answers one of the questions, which was, is there any spatial control over what, control over what this thing is methylating, right, FTO?
Right, yeah, that's a very interesting question. So in mammalian system, we have a hypothesis because it works on the stem cells. You can think about the stem cell as a high energy ground, and then during differentiation goes down, right? During that process, the chromatin is more closed, but the presence of FTO opens local chromatin. So during this compressing state, you still need to open lots of genes that are good for development. So, you can't do it with histone or DNA methylation because that's it's trying to close the chromatin. Use RNA to do the job. And we believe in plant system, what we had is a state in Amazon where there's just the sunlight and lots of water. Then plant gets into this epigenetic state, it just grow crazy, right? And we believe because of the demethylation, we hit on that sweet spot that we open up the chromatin and then plant just grow crazy and they locked into that epigenetic state. We have a lot of unpublished data supporting this hypothesis.
Thank you, John. One more question, which is there from Peter Hammack, which is a really interesting question. He says, was there a change in nutrient demand between the FTO and non-FTO plants?
Yes, that's a very good question. So there's several layers of that. First of all, the grains we isolated has the same sort of nutrient or same sort of context as the Y-type control. And yes, if the plant grow faster, bigger, as we showed in the lab, if you keep supplying nutrients, it will just grow bigger, right? In the real field, we give the same amount of fertilizers, nutrients as the Y-type control. In this case, we get a 50% increase. So it maximizes the potential. If we supply more nutrient, it will probably grow faster or grow bigger.
Don Ferruccio asks, is there a downside to FTO plants, like say decreased competitiveness or decreased pathogen resistance?
That's a great question. This is the first question. When I saw this phenotype, I was like, wow, why nature does not take this, right?
Yeah.
I think there are several layers of answer to that. One would think we'll track animals, so these plants get eaten up. Pathogens, surprisingly, these plants are a little bit more resistant to pathogens from preliminary results. It's not, I think it's just they're more robust. Now, what's the downside of this? One hypothesis we haven't tested is that, we have other results in different plants. We showed that the metabolism is accelerated or elevated. So some of the products, we actually get more per gram of plant compared to the wild type control. So the potential downside, I think, just my personal opinion, is if you have a plant which has a half-life time of 20 years, this may shorten that to 12 years, right? Grows faster, metabolism goes faster, but then maybe have a shorter lifetime. For food, it's not a problem because we harvest every year. For trees and grass may not be a problem as well if we have a faster turnover. But during evolution, maybe that's the reason why they're not selected.
So we have a couple of related questions. So Gary Patrick asks, will FTO plants fix more carbon? Will it help reduce global warming? Someone else said that, could you use this in bamboo or something, other larger plants that will help carbon fixation? So
yes. Actually, we had a meeting with a lot of folks, and there are lots of great ideas. There are all kinds of great ideas we're currently thinking. For instance, even in economic trees, the plants that, not only you produce whatever you need, but also you grow a higher biomass. Whenever you have a higher biomass, you fix more carbon, right? So definitely this is something, it fixes more carbon for sure. The biomass increases. And this is something I'm extremely interested in.
Yes. So GLU asks, in plant applications, it is clearly a GMO technology, which would be regulated for public acceptance and environmental safety concerns. Are there any strategies to make it non-GMO?
Yes, we've already made it a non-GMO. We're in the process of IP this. Jing has a great question. We did it in GMO just to, show it works, but the fact is it doesn't need to be there. We have a paper, so we're going to submit in a couple of years. This is the science part is even more fascinating than what I showed here. It's actually incredible that we don't need FTO there. Actually, we completely don't need FTO. We can still have this crazy growth phenotype. That's the most fascinating part.
Oh my God, we have so many questions about the FTO rice. So will FTO rice be safe for people to eat, Chuan, from wrong year?
I think I don't know. I think the students there ate them already.
Do studies.
We analyzed everything of the rice compared to the white type. Essentially no difference.
Yes. So Christine Romano asks, you mentioned that overexpression of FTO in plants opens up chromatin, allows expression, more gene expression. Are there any specific genes that are overexpressed? And I think this is a fantastic question.
Right. So as I showed, this is basically the science part we wish to continue to explore.
Yes.
Majority of the genes are upregulated. Something like 80–90% of the entire genome, their expression is elevated. Specifically, the genes involved in photosynthesis, in differentiation development, these genes are upregulated. I talked to plant biologists. I think there are several ideas. One is that when you have a root development, right? The root is more like a stem cell development. When you have a deeper, larger roots, that turns to also send a signal into the up plant to tell the plant to grow. So the system could come from the root, but also on the top, there also might be effect from the shoots and other places. So this is also a answer to Jean's question. Once you get your root or at the seed level, once you lock into that epigenome state, what we realize, and then you start to grow crazy in roots, Now you don't need the FTO. Once it lock into that state, it just grow.
Okay, in the interest of like not making this a botany talk, I'm going to look for some questions which are related to the first part of your talk, Chuan. And this is from Tejas Patel, who says, very fascinating talk, which I agree. What are the downsides to YTHDF1 blockade, as PD-1 inhibitors have also shown surprising side effects?
We have not, this is a great question. This is a great question. I've been thinking about this for a long, long time. So YTHDF proteins, including DF1, seems to be repressor of immune response, right? The question is, what's the natural role of YTHDF1, right? Why do we have this function? It's not there to help us fight tumor. That's not the reason, right? So one can think about during early development, the tissues grow very fast. This may be a way to make sure we don't attack, the immune system doesn't attack a fast growing tissue, right? And yet, the knockout mice did not show any immuno effect. So I'm still surprised and puzzled by this. So far, we have not seen this adverse effect, but I do want to mention that if you knock out the methyltransferase, you wipe out the entire methylation. The YTHDF1 works on a subset of the methylation, but if you knock out the entire methylation in T cells, you do get IBD diseases, for instance, these autoimmune diseases.
So in fact, Matthew Nida asks another very interesting question. He says, do you think that there are other more specific RNA methyltransferases and demethylases?
Yes, we have another paper we published this year in mitochondrial. For instance, we found another demethylase. Yes. And that originally we thought works on tRNA, but no, it also works on pre-mRNA. In a way, it works on this mitochondrial pre-RNA, and it doesn't work on the mRNA versions, it works on tRNA to regulate the processing of the entire RNA. So in human mitochondrial, there are two RNAs that are first transcribed. That is get chopped off into many, many pieces. That chopping process is regulated through this methylation and demethylation.
I have been given instructions that I'm allowed to ask you only one more question, Chuan. So I'm going to pick a fun question. And this is from Rosemary Safranek, who asks a question which I also want to know the answer to. And it is, with so many interesting ideas, how does Professor He think about how to spend his time?
That's a great question. Well, I want to say that it's really, I'm here to give this talk, but it's really work of my lab, which is a lot of people, Yamiona and I share the floor. They work so hard. I came into my office, they're still there working. My collaborators, my former students, there's just not one single person's efforts. It's really collectively people work with us, help us. It's a group, it's an effort from everybody.
But how do you spend your time, Chuan?
I think the key is you work on something different. I think the key part is you choose something what not to do. You choose to do things that you think are really valuable.
Absolutely, absolutely. So thank you, Chuan, for a fantastic lecture. And so with this, I'm going to hand over to Andrea.
Oh, thank you so much, Professor He, for sharing your work with us. This is fascinating and it's going to help so many people. It's amazing. Thank you, Professor Krishnan, for being here to extend the conversation so thoughtfully. We also invite you to listen to Big Brains, a podcast featuring the pioneering research and pivotal breakthroughs at the University of Chicago and beyond. In episode 74, Professor He discusses immunotherapy, biomass, climate impact, and much more. So visit bigbrains.uchicago.edu or find Big Brains wherever you listen to podcasts. Thank you again. We'll see you soon.